Hi everyone. Today's study tips are thinking about what's most important. So in anything we study, some things are more important than others. And it's important as you work in your college career to identify these high priority items and build your studies around them. So notice where the instructor spends the most time or space. If we talk about one thing for 1 minute versus for an entire class, there's going to be differences there. Pay attention to those main concepts then. And if steps are given, know them in order. This is really important for the mechanistic things we do in physiology class. Finally, you should practice. Answer end of chapter questions. Try to teach the concepts to each other. And practice writing out explanations of the main concepts and the mechanisms. Today we're working on chapter 18, the first part of the digestive system. And probably most of you know this, right? The functions of the digestive system are motility, which is moving and mixing food. There's secretion, and so our bodies secrete water, buffers, enzyme, and mucus into the system. Digestion, of course, catabolism of foods into their smallest units, usually by hydrolysis, so we'll talk about that process today. And this includes turning carbohydrates, breaking down carbohydrates into monosaccharides, breaking down proteins into amino acids, and breaking down fats into glycerol and free fatty acids. Here's what hydrolysis looks like. When you want to break down or catabolize a big component like carbohydrate, you need to use water to break it down. So here's maltose, a disaccharide, di meaning two, so there's two sugars in this maltose molecule. If you add water to it and make that reaction happen faster with carbohydrate digesting enzymes, you end up with glucose and glucose, so two monosaccharides. So that water is responsible for splitting maltose into two. And you can see that because water is H2O. And so we add a hydrogen to one glucose molecule and an OH to another. And that's how we get to split. So enzymes are needed to split apart molecules into their monomers so that we can absorb them into the bloodstream. And remember that enzymes are going to speed up reactions. They're not going to cause reactions, but they make them happen so much faster that it makes sense for them to be used in our body. If we didn't have enzymes, we wouldn't be able to complete all of these digestive and metabolic things we need to do. So in this case, the enzyme is using water to split, and that's called hydrolysis. So we're going to see hydrolysis, this way of water breaking down molecules in multiple ways. So here's the protein. Here's a protein. It's actually a peptide, which is a portion of the protein molecule. Again, using water and a protein digesting enzyme to break it down into two amino acids. We can do it with lipids, fat and water. In this case, we need 3 water molecules because we are getting out of this reaction 3 fatty acids and 3 glycerols. Okay, so next we're going to talk about how we don't actually absorb these components directly into the blood. We absorb them into the lymph system first and then into the blood. Okay, so absorption is another major function of the digestive system. Nutrients move from the lumen to the blood or lymph. We have storage and elimination from the large intestine, so it's getting rid of those waste products after digestion has occurred. And the digestive system also acts as an immune barrier. So the GI tract is a tube from mouth to anus, so technically it's outside the body. It's a continuous tube from here to here. So that means that anything in this digestive tract is outside the body. Okay? So this tract protects us then from things that are in our intestine. And some of the ways in which they do that is through the use of simple columnar epithelium, tight junctions, and lymph nodules. Okay, so there's a reminder of all the anatomy in the digestive tract. Okay. In addition to the actual tube itself, going from esophagus, stomach, small and large intestine, you also have all these accessory organs, right? The liver and the gallbladder, the pancreas in here, all of which have different jobs in order to complete that role of digestion. So go ahead and review these on your own time. Okay. Next, think about is the GI wall anatomy. So let's test ourselves. Write it down in about 4 minutes. Place these layers in the correct order from outside to inside and describe the tissue and function of each layer. So pause the video here, write this all down, and then come back. So here is our digestive tract. Got the serosa on the outside, a layer of longitudinal muscle, a layer of circular muscle, and then you've got submucosa and mucosa in there. Okay. The arteries and veins in the mesentery are also going to be associated with this and look almost continuous with the serosa, right? But not quite. Here's another way to look at it, a cross-section of the small intestine with the serosa on the outside. Then you have your muscular layers. Then you have your mucosa and your submucosa. Sorry, submucosa and then mucosa if you go from out to in. And note that inside here we have all these villi, right, which increases surface area for absorption. So let's start in the mouth. In the mouth, we have mechanical digestion by mastication. This is chewing. So we're breaking things down by chewing them. Saliva is also a really important component in the mouth. We produce about 1 to 2 liters of saliva per day. And saliva contains amylase, an enzyme which allows for digestion of starch. So we're starting to digest carbohydrates in the mouth. I want you to note that we have both a chemical digestion happening in the mouth as well as a mechanical digestion. Mucus in saliva lubricates food, and there's lysozymes in there to destroy bacteria and prevent dental cavities. Dry mouth is particularly bad for oral hygiene. So the drier your mouth is, the more bacteria can grow and the more damage you can have to your teeth. There's no absorption of food happening in the mouth, right? We're just beginning digestion both mechanically and chemically. Then that bolus, that chunk of food mixed with saliva, has to travel down the esophagus. So what happens is the esophagus is made of these muscles, and you get peristaltic waves. So waves of muscular contractions that push that bolus down from the mouth into the stomach. Okay. And here we are entering the stomach here. So the swallowing reflex that we have as a reflex pushes food into the esophagus. And then the peristalsis moves the food down the esophagus. Now there's multiple sphincters along the way that help control what moves when. So we'll talk about those today too. And the first one I'll talk about is this gastroesophageal sphincter, which prevents acid reflux. So you push this bolus down into the stomach, but there's a sphincter in the esophagus that's going to prevent acid reflux. Changes or problems with the sphincter can result in feeling acid reflux and actually getting some of the acid back into the mouth, which can be very dangerous. The esophagus also secretes mucus to help push that bolus down. Some fun facts about the mouth. Every person has a unique tongue print. But a not so fun fact is that by age 60, most people have lost about half of their taste buds. And so the question has been, maybe kids are picky eaters because they taste so much more than adults. And this might also be why kids like to have their food separated out as opposed to all together like some adults do. Okay, so now our food's in the stomach, and we can talk about the stomach functions. So the stomach functions to store food. We get the beginning of protein digestion here with the use of pepsin. We have a little bit of absorption in the stomach, but the only thing absorbed in the stomach is alcohol, aspirin, and water. So not much is absorbed in the stomach, but we get the beginnings of that. The stomach also secretes gastric juice from the gastric pits. And when you add the juice and the food or the bolus together, it's called chyme. So chyme is the mixture of our food that came as a bolus from the mouth and the mixture of gastric juice from the gastric pits. So now you have mixing. Peristalsis, so again, muscular contraction, about every 20 seconds when there's food in there. That's going to mix the food and the gastric juice together. And finally, the stomach empties. You get about 1 tablespoon of chyme through the pyloric sphincter. And this is slow to allow time for absorption. So pyloric sphincter at the bottom of the stomach, that's going to allow emptying. So we need to first look at the stomach cells and their secretions. And remember that we have this lining here with these gastric pits. And so you can see there's different cell types along the lining of the stomach that are going to do different things. Okay, remember we still have these same layers. We've got submucosa and mucosa here supporting this. So note we've got mucus cells, mostly towards the top here. We've got gastric glands, parietal cells, and chief cells. So 4 different cell types that are going to have different roles then in the stomach. Okay. The goblet cells produce the mucus. The parietal cells produce the gastric acid. And note that the mucus is really important because it protects the stomach lining, right? You're secreting an acid, a gastric acid, that is between pH 2 and 3. So it's really acidic, that normally would be breaking down cells. So the mucus protects the stomach lining from the actions of this acid. Chief cells produce pepsinogen, which is a protease precursor. So a precursor to an enzyme. Remember that EASE? Protease is going to break down proteins. D cells produce somatostatin, which inhibits acid secretion. And then the opposite, the G cells produce gastrin, which stimulates acid secretion. So we have 2 hormones here, gastrin and somatostatin, that control acid secretion. You want to secrete acid when you have food present in the stomach. You don't want to increase acid when you have nothing in the stomach. And finally, we have these ECL cells. ECL stands for enterochromaffin-like cells that secrete histamine and serotonin, and these stimulate blood flow in the stomach. So lots of cells working together to break down things and start to digest things in the stomach. The parietal cells, so the ones that make gastric acid, are going to pump hydrogen ion into the lumen via hydrogen ion potassium ATPase. So you got a pump here, right? Hydrogen potassium ATPase. And chloride then is going to follow to make HCl. So HCl is our acid, our gastric acid here. Okay, so that's the mechanism of how they make that, is by using this pump. Okay. So now you're probably thinking about gastric ulcers, which are very painful and dangerous, and relatively common too. So an ulcer is an open wound, and when the mucus layer that protects the stomach lining breaks down, an ulcer can form. And usually this can be caused by infection, particularly by a bacteria called H. pylori. And this is actually a relatively common bacteria, and that's why we see relatively common ulcers. It can also be caused by high doses or prolonged use of NSAIDs, especially if combined with alcohol. And so we will talk about destroying of the stomach lining when you have this kind of combination, and that can cause ulcers as well. So essentially then you have an open wound in the side of your stomach, which can cause leakage of things like gastric acid into the body tissues. Okay, so once food leaves your stomach, remember you get that 1 tablespoon per time coming through that pyloric sphincter. Now you have movement into the small intestine. The small intestine is made up of the duodenum, the jejunum, and the ileum. And this is where you have most digestion and absorption taking place. We're moving nutrients from the lumen to the submucosa into the blood or lymph tissue. And so we're actually getting absorption of most products here then in the small intestine. We get about 9 liters of absorption per day. 7 liters of that is digested juices. So we eat some food, but we also are absorbing then most of our own products that we're making. The surface is villi and microvilli, which is folded to give us increased surface area for absorption. And a brush border of enzymes and microvilli for final digestion. Okay, so again, we're looking at the submucosa and mucosa. In this case, the small intestine. We see the villi here, these folds, increasing surface area. And so within them, what you'll note is that we've got a capillary network in there, okay, that's surrounding this lymph vessel. So projecting into each villi, there's a lymph vessel surrounded by a blood supply. And so when things are absorbed, they can enter the blood or enter the lymph. You'll note that there's more goblet cells here, on the lining of the small intestine, okay, as well as a simple columnar epithelium. And we actually have, remember this is very long, so we actually have some segmentation in the small intestine for mechanical digestion and to slowly move the chyme. So we only have circular muscles here that are pinching and creating these waves to move chyme through that small intestine. Here's a picture from our anatomy class, obviously pre-COVID because not everyone's wearing a mask, where we're dissecting cadaver intestines. And so when you did this, you remember how long these intestines are, right? The small intestine has the surface area of a tennis court. So the average small intestine in a human is 7 meters, 22 feet long, and it's all folded up inside your body so as to increase surface area. Once chyme leaves the small intestine, it moves into the large intestine, but by this point, you've had a lot of absorption. So a lot of things have been taken out of that chyme at this point. So the large intestine is mostly for storage. We get a little bit of absorption here. Motility is caused by what's called a haustral contraction, about every 30 minutes. And most absorption of happening at this point is salt and water. Okay, so we've absorbed all the nutrients from that chyme, and now we have absorption of salt and water in the large intestine, and that produces feces. And then there's a defecation reflex to get out these feces. You have an involuntary internal sphincter that's controlled by smooth muscle, and a voluntary external sphincter that's controlled by skeletal muscle. So you have two sphincters have to then open up in order to actually get out that feces and defecate. Showing you some more anatomy of this. Here's this ileum coming in from the small intestine. And first you have the cecum, which is sort of this blind pocket here, and the appendix. So you can see why you might occasionally get infections here is that this appendix is a blind pocket, and so bacteria can hang out in there and cause problems. Whether the appendix has a function has been a question for a long time. It turns out it does actually seem to have some immune function. So not really digestive, but it's not completely useless. Okay. And then you have the material moving into the cecum, the ascending colon, the transverse colon, the descending colon, and finally sigmoid, and then out the rectum and the anal canal. Okay. Start thinking about things like colon cancers. Certainly you can have cancer along any branch of the colon, but these are going to be different a bit from rectal cancer here or anal cancer. So it's going to depend on which tissue the cancer is in, but turns out the mechanisms are also slightly different in terms of that cancer formation. Okay, so really importantly and for function of your large intestine is fiber. Fiber in the diet stimulates mucus secretion. So this prevents constipation and allows you to move that feces more easily through that large intestine. And in this large intestine, we have commensal bacteria. These are good bacteria that digest some cellulose, short-chain fatty acids, and actually make us some vitamins. So these bacteria are not infectious. They are not dangerous. They are good and required because we need things like vitamin K and some B complex vitamins. These bacteria in our gut are really important to maintain health. So some questions just to think about or look up are what are some good sources of fiber and what foods have good gut bacteria? So how can we make sure to support and provide bacteria to our gut? Okay. Finally, I provide 2 questions for you that you can look at and think about, write down some answers to, and then if you want to come see me in office hours, we can talk through these questions. So the first one is about someone with an ulcer. And this person with an ulcer has been prescribed 2 medications by the doctor. Okay? The first medication is called Tagamet, and it prevents hyperacidity in the stomach. The second medication is antibiotic. So your task is to explain why each of the medications would be helpful. Why were they both prescribed for this person that has an ulcer? Okay. Second question is about Michael, who went backpacking in a pristine mountain location for a weekend with his friends. 2 weeks later, he went to the doctor with symptoms of diarrhea, dehydration, abdominal pain, and cramps, and he was diagnosed with giardiasis. So how do you think Michael became infected by this protozoan Giardia? If Giardia colonizes in the duodenum and jejunum, what important processes is it inhibiting? And finally, how can backpackers prevent this? Okay, so write down answers to those questions, think about it, do some research, come back and see me about it, and we'll talk about digestion more in part 2.